Hi, biology students. So you should have watched the first part of my videos, and pardon me if I have to stop. I have my toddler here. Anyway, you should have watched the first part, and I told you about your journals and when the test is, and now I have your study guide. See, study guide. Okay, so you should have this. I wanted to go over it with you really quickly so that you could see. I could just clear up anything in case you had any questions that the sub couldn't answer. So the first part of it, it has definitions, things you need to know. So you need to know independent variable, dependent variable, control group, experimental group, hypothesis, theory, law, observation, conclusion, inference, precision, and accuracy. So precision and accuracy, remember we talked about, we talked about that when we did our station lab and we had the pictures of the bullseye. And precision was how well the person hit the center of the bullseye, which, I mean, well, you know, center of the target called the bullseye. And then, oh wait, excuse me, that was accuracy. Accuracy was whether or not they hit the bullseye, and precision was how many times they actually hit the same mark. So in the lab, you can think of it as accuracy is whether or not it's actually true or right, and precision is whether or not it's repeatable. So you could think of this as a balance beam. Maybe you're getting the mass of something. The fact that you actually measured it right would mean that it's accurate. And if you did it over and over and you got the same result, that would be precise. If you did it over and over and your results were different, then that would not be precise. Okay, next. Safety. You have to know all the safety guidelines. We already took a quiz on that. Everybody made an A or a B, so I don't expect you to not know safety. Just remember there were some weird little things on the safety quiz. So, like, for example, how do you hold a scalpel? A lot of people have said point it down, but it's actually hold it as you would a pencil. And this was something that was not in our safety guidelines. But hopefully that will not be on the test. Okay. Next, scientific method. This is what I wanted to talk to you about. Basically, I showed some of you this, and I don't know if I showed all of you, but I do have a, please don't, no. In your journals, you should have something about experimental scientific method, and it should have something like dry mix. And dry mix. Let's see, can you see it? Dry, D-R-Y, dry, and then mix. Okay, so the D in dry stands for dependent variable. The R stands for responding, and the Y stands for y-axis. So why should you care? Well, a lot of people have trouble differentiating between the dependent variable and the independent variable. And oftentimes the variables are referred to as either manipulated or responding, and you're not really sure what they're talking about. So this is a good acronym for you to remember that. So dry mix, D, dependent variable, R, responding variable. So the dependent variable is also known as the responding variable, and it goes on the y-axis, which points where? To the sky. On your graph. Next, you have mix. So manipulated is what the M stands for, and manipulated means the thing that the scientist changes. So I picture the person with their fingers in the cookie jar. Independent is independent variable is the manipulated variable. X is the x-axis, which is horizontal. Okay, now let's move on. So I have the scenario. Scenario 1. I have Miss Weiss wants to know whether or not the drug that she's created will help her students to be successful on their first test. The drug is called Remember All, and Miss Weiss suspects that it helps students to remember anything that they've studied. So for test 1, Miss Weiss gives her 4th and 5th periods the drug Remember All. Periods 6 and 7 get a placebo, fake pill made of sugar, which is not the actual drug. Miss Weiss uses the test scores from period 4, 5, 6, and 7 to see if Remember All works. When she graded the test, Miss Weiss saw a 20% increase in test scores in periods 4 and 5 compared to periods 6 and 7. Okay. So, what is the independent variable? Well, that's the thing that the researcher is changing, which is why it's known as the manipulated variable. Hi. So what am I changing in this experiment? Okay, I'm changing the drug, right? Either they're getting a drug or they're getting a placebo. So that's what's being changed, okay? My responding variable is what? That's what's going to change in response to my manipulated or independent variable. So what am I expecting a change in? Remembering and test scores, right? Okay, so those are those two, right? What would have my hypothesis have been? Okay, so since you have to figure that out, my hypothesis probably would have been, you know, if I give my students Remember All, they will do better on the test. Okay, so usually your hypothesis is what we call an if-then statement. So if I do this, then this will happen. So try to remember to phrase your hypotheses as if-then statements. Okay, next. Experimental group versus control group. Which was my experimental group? That would have been periods 4th and 5th because they got the drug. Periods 6 and 7 got nothing. They got a fake pill. So that would have been the control group. Okay, so experimental group is the one that I'm experimenting on. Control group is the one that I'm comparing my experimental group to, that I'm purposefully not changing anything so that I can have a comparison. Okay, let's see, what else do you need to know for this one? Conclusion. If I saw a 20% increase in test scores of the students who took Remember All versus the students that did not, what could I conclude? I'm going to let you think about that one. So then you finish scenario 2 and 3 on your own. Characteristics of living things. We haven't gotten to that, but you should have read a little bit about it yesterday in your reading. We have done science versus non-science, so I expect you to be familiar with that. It's also in your notebook. Then there's DNA, which we have not talked about, so find that in your book and answer those questions as best you can. Whatever you can't finish, I will assist you with and teach you when I get back. Macromolecules, I haven't gone over that either, but try and find it in your book. Use your index, your table of contents, things like that to find these things, okay? Don't expect anybody to just know where it is because it's kind of spread throughout the book, okay? And that is it, I believe. Let me make sure. Study guide. Yes. Okay. That's all. So your study guide is due. I'm going to check it on Friday. So you'll work on it today in class. And then when I get back on Thursday, we'll talk about it. Then Friday, we'll go over it as best we can. And I will check it to make sure that you did what you're supposed to do and so that you can get credit for your work. Come with your questions when I get back, and I will see you Thursday, hopefully. Bye.